We have Aaron Epperson. He's worked with bears for 7 years, including time with the U.S. Fish and Wildlife Service as a wilderness ranger at Pat Creek Brown Bear Sanctuary and Admiralty Island, with the Oklahoma Department of Wildlife as a research technician studying black bears, and worked as a bear management ranger at Sequoia National Park. Aaron is currently a Wildlife Technician 3 in Palmer and is excited to share work from this groundbreaking collaborative project. Take it away, Aaron.
Well, thank you all for joining us for this evening's exciting bear research talk. The team here consists of Jeff Stetz, myself, Sarah Rockenstein, technician, Trista Sims, I'm also a technician, and this project was done in collaboration with ATRIC, U.S. Fish and Wildlife Service, U.S. Geological Service, multiple state agencies, multiple captive facilities, and what we're trying to do is develop a molecular clock to be able to age bears from their DNA. So let me see if I can get my slideshow to work. There we go. In today's talk, we will discuss why we care about age, how we estimate age, and a little bit about DNA. We're going to talk about epigenetics and our research project. So what I want to do first is just ask everyone how old you are. And if you could answer that poll question, that'd be great. And while you're answering it, I just want to let everybody know I took this picture at the McNeil River, which is in western Alaska. And it's an awesome place if you're into bears and bear viewing. Even if you're scared of bears, I really do suggest you go here because you won't be afraid of them anymore. They might be 5 feet away from you, but it's really cool just to see bears not influenced by the fear of humans that they often have in a safe environment. And it's cool to just get to see bears be bears. It really does help you gain a great appreciation for them and get really cool pictures. So I really suggest you check out the McNeil River if you get the opportunity. There's 10 people that were 23 to 35, 9 that were 35 to 45, 12 that were 45 to 55, 8 that were 55 to 65, and 11 that were 65 to 75. And so that's our age distribution of this audience. And that was an easy thing to get because we were just able to ask. It's a lot harder to get from wildlife because we can't ask them. So what we try to do is evaluate population demographics. Population demographics define a population. Population abundance is one of them. It's how many individuals are in the population. Density is how many individuals are in a given area. Age structure, or how many individuals are in the population. This is used to define cohorts. Cohorts are grouped by age, for example, those that are too young to reproduce or are able to reproduce, and the final age group would be older or less reproductive in the population and don't add as much to the population. Survival is a population demographic, and reproduction, and those are both tied to age. As animals age, these rates tend to drop. Transients or dispersing animals can be linked to age. For example, subadult bears will disperse in search of home range. Birth rate is how many offspring an individual can have and at what frequency. Mortality or death rate of individuals can be called survival. Sex ratio, which is how many males to females are in the population. And with this information, wildlife researchers can manage populations accurately and sustainably. Meaningful insight into population trends is key for understanding the population performance and trajectory. And as a management agency, we have a mandate to try to use the best information and best methods that we can to try to understand populations of wildlife. So why do we want to know the age of animals in a population? Well, we want to try to define that population. It's super important, and often it's very difficult to do. For example, does density estimates include just adults, just dependent age animals? Dependent age animals are cubs with adults that are dependent on their mother, or does the estimate include everyone? If we don't know for sure, we have to make assumptions, and we all know what that means. So differences among age classes likely introduce noise or more variability to the data, resulting in less precise estimates of survival. So we want to be as precise as we can when trying to figure out the age of these animals. Population growth rate is one number to summarize what's happening in a population. This is the formula we use to calculate that. It's births. It's the BIDE formula, births plus immigration minus deaths minus immigration. So variation in life history traits affects population growth, and patterns differ dramatically based on life history traits of the species. So in this graph, we're looking at humans up here on the top in the upper right, and we see generally that humans tend to have one baby at a time, and when born, most of those babies are going to survive to be adulthood and die at old age. We tend to have extremely high survival rates until we reach that tipping point at old age. And in contrast to the humans is oak trees. They have 10,000 little acorns that hit the ground per year, and most of these acorns are not going to survive. But if they do, those that reach maturity are going to live very long lives into the future. So that's two different strategies there. This graph shows that body size can affect growth rate. So the larger the animal, the longer it tends to live. So we're looking at elephants in this graph. This graph right here on the bottom right is how body size can affect reproductive rates. The larger the animal, oftentimes the fewer offspring that they have. Whales are a good example. They might have one calf every 4 years. So let's take a little bit closer look at population growth rate. Growth rate reflects the values of the BIDE equation. Immigration and emigration can be affected by age, but the point here is to look more at the reproductive success. So on here, we're going to look at age-based survival rates and reproduction. This graph right here shows reproductive success increases with age and plateaus, and then it begins to decline. This graph shows the survival probability of an animal. It increases, and the animal we're talking about is this reindeer right here. It increases with age and plateaus until it begins to decline. Older populations reproduce less, and that can confound. These patterns work together to define a population's growth rate. The most important factors of population growth rate, therefore, often directly are linked to the age of animals in that population. This can cause additive effects. For example, at prime age, both survival and reproduction are at their peak. And when these are combined, there are more animals on the landscape. Knowing these values for individuals when applied at a herd level can project population performance of that herd. So the age patterns can affect offspring survival. This is called the Lansing effect. It was first noted in humans by Alexander Graham Bell in 1918. This graph depicts humans, and it shows higher mortality of offspring in older parents compared to younger parents. At a population level, these patterns can have strong effects on population growth rates. So the graph on the bottom here shows a population. This triangle on the left side shows a lot of individuals in the young adult age class. And as these individuals advance into the adult age class and they're reproducing, they're going to add a lot to that population. Over here on the right, we see a shrinking population. There is a lot of adults, and there are fewer young in the cohort. So, oops. So that indicates a shrinking population because there, for some reason, are not as many offspring being put into the population, and we see a shrinking population there. So researchers use these population bubbles to predict the future performance and trajectory of a population. We have a few different methods of determining age in wildlife. One of the standard ways is phenotypical aging, or how it appears, what we can see and estimate. Phenotypical age determination is used to age animals. For this, there are things we don't know that affect what we are measuring. So some of these techniques include antler size in moose from aerial survey. We're going to look at the moose and see how big the antlers are and classify it into an age class. Some of the classifications there we'll use are small antlers under 30 inches, medium antlers up to 50 inches, and antlers over 50 inches. And if you're over 50 inches, you're a mature male bull. We can also look and see if it's a cow or a calf, and then what size that calf is to figure out what year of life it's in. And that's all done from an airplane. And so we do the best that we can do. The body size of polar bears can be evaluated in an aerial survey, and they're looking to see how big that polar bear is, and they're going to classify that polar bear into an age class. Horn annuli is used to determine the age for goats. This can get a little bit difficult when they get up over 8, and because the annuli just start to stack in on the base of the horn. Another phenotypical way we can age animals is the growth rings in teeth or earplugs for whales. That usually requires dead animals to do. And we can also count annuli on the dull sheep horns. So at a young age for bears, we can phenotypically age them by evaluating their skull size. This is often done off of a harvested bear. And what we're looking for there is overall skull width and length. And then, oh, yeah, I have a skull right here. So when we're going to age a bear to see how old or young it is, and we want to make sure it was mature, we measure it from here to here, and then from here to here, and that's the total skull width. And if that's coming in small, that's a big indicator. And the next thing we'll look at is the eruption angle of this tooth right here. When the skull is fully developed, this eruption angle is here. When it's not, it's a little bit further out. And then we're also going to measure the length of this tooth right here. And with those, we can tell what age class that bear is, or we can at least tell that it's not fully developed and not in the adult age class. And so those are some examples of phenotypical aging for some animals. We can also evaluate the wear pattern of teeth. So deer family, this includes moose. We can look at the wear on their teeth. It's evaluated to determine age. This can be reliable, but it's dependent on regional differences in the soil type, the browse type, and the browse behavior of the individual animal. Moose in some areas show tooth wear much faster if there's glacial silt on the vegetation or in areas where plants have more silicates in them, and this increases the tooth wear. You can extract teeth from ungulates and send them into a lab, and you can count the cementum rings on them. They're cross-sectioned and the lab counts them, and you get an age estimate back from that. And that's traditionally how we've aged bears. From an extracted upper first premolar. A lot of times we take a tooth that's right here. This bear is missing it. He probably got aged. And so that's the easiest tooth to get. And you can get the bottom premolar too. It's just a little bit harder to get out. And so teeth from bears and ungulates are sent to a lab where they are cross-sectioned and they count the rings to estimate ages. There are some limitations to age determination with teeth. For now, it's the best way researchers and managers age bears, but there's been multiple studies that have indicated an error in this method. The U.S. Fish and Wildlife Service found young polar bears were overestimated and bears over 8 were underestimated. They compared cross-section tooth estimates to known age bears. So it's the best that we have, and it's a good an estimate, but it does have some error. So one of the things that we have to do in order to get these ages is handle animals, and that comes with some drawbacks too. It can limit your sample size. Samples from harvested animals are biased towards large, older males in trophy hunting areas because that's just what people are trying to harvest, like in Kodiak. It can get you a limited sample size. If you're capturing them to age them and pull their teeth, you're only going to get the animals you can find and capture. There's a high operating cost, and there's a lot of safety considerations when it comes to doing capture work. Capture work in Alaska requires aviation. Multiple personnel are often required to do these missions, and it can be dangerous not only for humans, but also for the animals. So those are some of the drawbacks of getting that data. We also have some ethical considerations of pulling the teeth. We assume that the bears don't really need that first premolar, but they evolved with them. So I kind of suspect that they might have them for a reason. And I also wonder what the rate of infection is when we pull a tooth from a bear or an ungulate. It causes them pain. You know, most bears I've captured, I just used a disassociative and a neuroleptic. There's not really any pain blocker in there until I administered a carprofen shot right at the end of that. And that's just a one-time anti-inflammatory shot. Ungulates don't have a lot of teeth, and when they're old, they're very worn down. So I think that the age of those teeth at that point really does matter. And aging from epigenetics could take advantage of a wide range of methods, such as scat collection or other methods like biopsy darting, and this could be more humane and more accurate. So sometimes we do have to pull teeth, and that is justifiable in a population estimate if we need to get it. But if we can find a better way to do it that's friendlier to the animals, that's what researchers, most of them really do try to aim to do. So we're always looking for better ways to increase the safety and comfort margins of the animals that they capture. And I know I've extracted a lot of teeth, and it'd be nice to see a day where we don't have to do that anymore. So one thing we do get from harvested animals and animals we capture is we now have a large database of tissue and hair that hopefully in the future we can use to age with this epigenetics technique. So getting into genetics a little bit, DNA research took off with the completion of the Human Genome Project, and it contributed a great deal to the knowledge of genetics. They mapped the human genome in the 1990s, and it cost millions. It's advanced a lot, and now humans can have their genome sequenced in an hour for $100. Advances in genetic analysis and computer science combined will bring on a new horizon of genetic research and applications of this research. So this is a graph right here, and it illustrates the increasing number of papers published using genetic sampling techniques since 1995. So it is really just skyrocketing there, and the acceptance of them is increasing, as is the use of them. So there's a lot of promise for it. Genetic sampling techniques, there's a lot of them. It's an ever-growing list of techniques for sample collection. This can include shed feathers. We can get DNA from scat. We can get DNA from saliva. This squirrel right here, it's a bite tag where the squirrel bites it, and now his DNA is in there. Urine, there's a tiger right there. The polar bears, they're getting DNA from polar bear prints. We can get it from the hair of bears that rub or any other animal. There's also environmental DNA. You can get it from the air. They're starting to do that, believe it or not, or in the water. Some of the interesting water sampling techniques are used to detect invasive species. In Alaska, pike. So they have this backpack, they put it on, they sample the water, and it tells them the occurrence of an invasive species. So you can get it from the whale snot. That's pretty cool. And so there's a lot of different ways that you can get DNA these days and use it. So I want to start this portion. We're going to get into epigenetics. I just want to say I'm not a geneticist, so bear with me. So how can we use genetic sampling to determine age? To find that answer, we'll have to look at a growing field called epigenetics. Epigenetics is the study of how behaviors in the environment cause changes in the way our genes work. Epi means on top of. That's what the Latin is, or above. So epigenetics is outside or above the genome. Your epigenome does not change your DNA sequencing, but it absolutely determines how your body reads the DNA sequence. You can think of it kind of like a computer. Your genome is the hardware, and your epigenome is the software. And the software is telling the hardware how to function, when to function, where to function, or in this case, what genes to express. Genetic changes alter which proteins are made, but epigenetic changes affect gene expression by turning genes on and off, like a light switch, or how much they are expressed, like turning a dial. So epigenetic studies go back to the 1950s, and we're going to dig a little bit further into it here. There's two common types of epigenetic changes that affect gene expression. One is histone modification, and the other is DNA methylation. The first, histone modification, is a protein that gives structure to a chromosome, and a combination of different molecules can get attached to the histone tails so much that they may squeeze the proteins and prevent the activity of the DNA that is wrapped around them. And so it hides the DNA from the cell because it's wrapped tightly and the cell can't use what it can't see. The other form of epigenetic change is DNA methylation, which is what activates or deactivates certain genes. Adding methylation markers to certain DNA bases prevents the genes close by from being expressed. And that's what our study is going to look at, DNA methylation. So one example of epigenetic change is the agouti mouse model. These two mice right here are genetically identical. However, one is obese and yellow and one is brown and thin. How does this happen if they're genetically the same? The answer can be found in the epigenome. These mice both inherited the agouti gene. When turned on, the gene leads to a yellow coloration and impacts the ability to regulate food intake, thus leading to obese mice that are prone to cancer and diabetes. In a genetically identical mouse where this gene was not switched on, we see a dark brown mouse, healthy, thin, and looks different. But what prevented the gene from being switched on in the brown mouse? Well, it was a small tag attached to the DNA called a methyl group. In this case, researchers gave pregnant yellow mothers food rich in vitamins like B12 or folic acid, from which they could make those methyl tags that silence the agouti gene. Fat yellow mothers gave birth to thin, healthy brown mice. This gene was activated by diet. The yellow mother gave birth to yellow mice that shared the same agouti gene expression, but only when the mother continued eating fatty, sugary foods. This predisposed her offspring to the same health concerns of the mother. So when the fat yellow mother switched to a healthy diet, her offspring then started coming out brown and healthy. And what's interesting is this really proves that what we do during our lifetime can affect our offspring. However, the offspring can change the inherited epigenome through things like limiting toxins and stress, proper diet, and so on. Another exciting fact is that we can alter our own epigenome through healthy choices, slowing down biological aging, which may prevent disease. It reconfigures the software on your genetic computer. So I just want to ask how old everyone feels. Do you feel older than you are or younger than you are? Great. Everybody, for the most part, is starting to show that they feel younger than they actually are. So that indicates to us that your chronological age is what we talked about in the first poll, but your biological age is how old you feel. So chronological age is the actual amount of time since birth. Biological age is the age our cells appear to be. So in order to understand how epigenetics can be used to determine age, we need to understand the two different ways to measure age. Chronological age is the actual amount of time since birth, and biological age is the age our cells appear to be. It includes not only elapsed time, but also extrinsic and intrinsic factors such as inherited epigenetic markers and epigenetic markers linked to lifestyle and nutrition. The methylation can be influenced by environmental, behavioral, and hereditary factors. These factors can affect our biological age, change the epigenome, and can become generational, and they can also be reversed. So like factors such as smoking can change the methylation of an individual's DNA gene expression These changes can cause disease and increase the biological aging of our organs. Parents and even grandparents can pass on the epigenetic change to their offspring. The offspring can then be more susceptible to the same health complications of the parent. And to say that again, it's fascinating that these effects may be reversed in both the parents and the offspring through healthy choices that turn off or on certain genes. So chronological age, how long you've been on the earth. Biological age is how old your tissues and organs are based off of factors. So methylation and age, this study is evaluating methylation. So with that in mind, we'll return to methylation. While research into methylation and its impacts on biological age from factors such as stress, diet, exercise, and exposure to contaminants has been widely recognized, there is increasing recognition that DNA methylation can help us determine chronological age. Researchers are looking for methylation markers that are independent of the biological aging. Biological methylation can vary greatly between individuals. That's why twin siblings could develop different diseases later in life. And we are looking for markers that indicate chronological age. These markers should be consistent across a given species. So Dr. Stephen Horvath developed an epigenetic clock in humans by charting 353 markers linked to methylation that change with chronological age and are present throughout the body. In a 2023 study, this study sought to develop universal pan-mammalian epigenetic clocks from DNA methylation profiles of 185 species. These clocks can then be applied to accurately determine the chronological age of an individual, but the clocks may need to be refined for a given species or even location. So here we have a study. It covered its bases very well, and it evaluated lots of life history events. The estimated age to actual age was within 1 year. The graph shows a tight correlation between actual age and the age from the DNA. So they generated 10,000 methylated arrays in areas of highly conserved DNA. They used 59 tissue types from 128 mammalian species, and ages range from prenatal to 139 years old, and they had a good weight distribution, and it was an impressive accuracy. We see the R value was close to 1. And so just briefly, the R value is a measure of how closely true age and estimated age are. For example, R equals 1 means the two are basically identical. And here, the median error means that on average, the estimated age was within 1 year of the true age. Some studies have found variation in methylation rates based on various factors such as sex of the animal, its body condition, stress, and even the age of the animal itself. This research shows a lot of promise, but to be upfront, there are a lot of factors that may affect these types of studies. Various studies have found this error. That's why it's important to validate our research on the species we're studying and in the areas we're studying. We need to be doing our homework on these species. This is a case study of canids. They used bisulfate conversion sequencing of 46 domestic dogs, 62 gray wolves, and they assessed the accuracy via cross-validation, and the final clock showed R was equal to 1. It's very accurate. We see a pretty close line there, and then the wolf R was 9.7. So the linear trend in this graph shows that the DNA age is closely correlated with the actual age of the animal, and the R value is 1, which means that it's very accurate. We're obviously skipping some of the details here in terms of methods and validation, but nonetheless, these were rigorous studies and they were peer reviewed. This is a case study on bats. They had a good sample size. They had 712 samples from 26 species from 6 families, a good age distribution, and they assessed accuracy via cross-validation. And so R was equal to 0.95, and flying fox is really close to 1. So again, very accurate. This was an early stage study that only looked at a very limited focus number of bears in Japan. And the genome they used was like 3 dozen versus 10,000. So in some other studies, they've used a lot more of the genome sequencing to evaluate methylation. And this is a good example of how quickly the methods are improving, providing better starting points for future clock development. And this study looked at how well molecular clocks worked with captive versus wild bears. And with other studies, the results are very promising. And it shows a tight correlation there as well. But they used blood samples from 34 captive bears and 15 wild bears. They started with just a limited number of sites on the genome. It was pretty accurate, but there was some variation there. And they found no effect of sex on the predicted age versus chronological age, but they found significant effect of growth from the environment via wild or captive bears. The difference between estimated and true age was a little bit bigger. They also found that the aging rate tended to decrease as animals aged in the wild, but could be a function of captive bears not hibernating. So we're going to switch gears now and talk about our ongoing research applying epigenetics to Alaska's 3 bear species. Each of these species is of tremendous value and both culturally and for consumptive uses and wildlife viewing opportunities, in addition to the roles they serve in supporting healthy ecosystems. As such, each of these species receives a great deal of effort and funding for conservation. So we have polar bears and brown bears and the American black bear in this state. This is the distribution of polar bears in Alaska. They have a big range. They're actually considered marine mammals. The females have a low productive rate. They begin reproducing at 4 to 6 until they're 20, and they usually have 1 to 2 cubs. So you could say they have 14 years of breeding, and they may only add, if a polar bear only had 6 cubs in her life, each litter being 1 cub, how many of those cubs survive? How many of those cubs make it to the subadult age class, and how many of those subadults make it out of that age class is even less. Subadult bears do tend to die at a high rate. And so it's really important to understand polar bear age distribution and their ability to reproduce. And they are listed as threatened under the U.S. Endangered Species Act. A cool thing about the polar bears is that a lot of species we see are less effective at rearing young at older age. Polar bears get better at it. They have less cubs, but they're better at bringing them into the subadult age class, whether that be they're better mothers, they've learned some lessons. The American black bear is the smallest and most widely distributed bear in North America. They generally live in forested species. They're much greater reproductive potential than the other 3 species of bear. Their populations are growing in a lot of different places. In Oklahoma, we did a study where age and sex distribution was very important. It was an expanding population. The core study area looked good. We started getting bears on the fringe areas. We did a mark recapture, and we found that the age distribution and sex distribution did not reflect that of a growing population. So we decided to not hunt them there. Brown bear is the other species in this state. They're the most widely distributed bear species on Earth. They're all over the place. They're very adaptable, super cool, really important to tourism in this state and trophy hunting, and they're also important to the ecology of this state. So we did a multi-year, multi-method study in Unit 13 to estimate bear densities and population growth rates. We added a collaborative genetic sampling study to estimate and explain variation in density patterns. And this study got 2,000 hair samples collected in 2022. It included 137 grizzlies and 143 black bears. It was a rare opportunity to compare inferences from multiple sampling methods. It was a large project, and it provided strong motivation to explore options for DNA-based aging. This picture right here on the bottom reflects where those hair traps were. Hair traps are just pieces of barbed wire that snag some hair as a bear walks past it to investigate whatever it's trying to investigate. So just the first step, and we're going to build on the results to address factors such as regional differences, but hopefully we can age those bears with epigenetics. What our study did, it was a collaborative effort, and what we wanted to do was get enough samples of known aged bears, not from a section tooth. So it either needed to be observed or known. This was a collaborative effort, and what we asked for was hair and blood from these bears. And we reached out to captive facilities, rescues, and researchers in the field. Some of these research people that we reached out to, like the Interagency Grizzly Bear Team, Department of Wildlife in Connecticut, and the people doing polar bear research, they've been studying those populations for a long time, and that allowed us to get wild bear samples of a known age. Otherwise, those samples would have been really hard to come by. So we're going to get those samples, and we're going to send them off to the lab to have them evaluated to the Clock Foundation, which is the nonprofit that was established by Stephen Horvath to facilitate the analysis and development of epigenetic clocks. One thing I got to note is that when we got into this and we started asking for samples, everybody was fired up. We had such great support. Everybody jumped in there and wanted to help, and we couldn't have done it without them. So big shout out to all of our collaborators. This is the preliminary results for polar bears. We sent 96 whole blood samples from bears captured in the Chukchi Sea from known age. They were captured as dependent age bears or cubs, and then these bears had later been recaptured, and blood was drawn from these bears. And some of them were also estimated from the annuli in their teeth from a cross-section tooth. The results have been promising. The top figure shows the correlation of predicted age versus tooth age. R was 0.85. That's pretty accurate. While the bottom shows a predicted epigenetic clock age versus known age. They knew the age of these bears, and R was 0.95. So they're going to further refine the model and results. They have 96 additional samples to contribute that are getting contributed by the USGS of a wider age range of known age bears that were all caught as dependents. And so the point here is when you know the age of your bear, your data is more accurate. So the future research could be bright for epigenetic aging. Researchers could use hair traps to gain insight into density, distribution, and age distribution questions without having to handle the wildlife. Conducting a mark recapture with only hair snares and game cameras could give insight into many questions regarding population biology. Currently, we are working to get accurate ages of individual animals. And once we build those clocks or develop those clocks, then we can apply them with wider application and start on other species and get data such as herd age class distribution for caribou. Ungulate researchers could build a clock for caribou. For example, biologists could biopsy dart a caribou herd from a helicopter and send the tissue into the lab. This would provide meaningful and accurate age distribution data across an entire caribou herd and could help researchers predict where the population is trending. Here at ADF&G, we have a great caribou research facility ran by Dr. Kristen, and we could potentially build a clock for caribou, and if anyone could, she could. So caribou management could potentially benefit greatly from this research, and caribou research is important right now because we're concerned about their population health. So why is knowing the age of animals important? Multiple reasons. We want to know population structure so we can give insight into population performance and contribute to the ability to predict future performance. And this graph right here is just showing that the caribou's performance and their ability to add to the population is changing over time. We want to be able to understand that and figure out why. So with that, I think that concludes it. I want to thank everybody for listening to the talk on epigenetics. These were some of our collaborators that we had that were super awesome in helping us get our samples. And now if you guys have any questions, it would be a good time to ask them.